And then each one of the, each one is trimmed some random amount. So that's sort of demarcated by this. And so it gets trimmed, and then they get joined together by random nucleotides. So that's sort of a third layer of the randomization. So that gives us what we call a naive B cell. And I've drawn this as a cartoon here, and this is the receptor on the outside, and also in a cartoon. So that's sort of drafting an antibody. And then once this B cell receptor meets its cognate antigen, something it binds to, it starts this process of somatic type of mutation and affinity maturation. So that's this Darwinian evolutionary process where the body introduces these mutations, like so. And the ones that make sort of a worse binding receptor, like this, get thrown away because they don't bind the antigen better. And the ones that bind it better are allowed to reproduce more. So you can kind of imagine this as, these antigens, we can imagine as being a bunch of blocks. And so the world is just constantly throwing blocks at you. And this process up here is basically making random keys. But these random keys don't always fit perfectly. They might fit somewhat well. Maybe they insert into the lock, but they don't actually turn. And so this process down here is sort of a process of randomly filing along in Darwinian selection on the key so that it fits and then it turns and opens the lock. So, just in case it wasn't clear from the previous slide, I want to emphasize this point. You have an enzyme that incorporates random nucleotides into your genome. That I find to be an unbelievable statement. There's this TDT that does this, and you also have an enzyme which is purpose-built to mutate your genome. This is called AIV, and I'll be talking about this more later. So, you also have these many organs that are designed specifically for evolution. So inside your lymph nodes, there's these things called germinal centers. And up here is a collection of cells called the follicular dendritic cells. And their job is to present the antigen. And then these are B cells that are highlighted in green. And you have these two different zones. So this is the reproduction zone, where they reproduce and mutate. And then they basically get tested up here. And then if they sort of pass the test, if they bind antigen well enough, then they get to come back down here and reproduce again. So if this seems interesting to you, just read anything by Gabriel Victoria, and I assure you, you will be amazed. So, okay, so we can sequence these things in high throughput now, and this has completely changed the field of adaptive immune research. So now I'm going to that was sort of my gee whiz, this is an amazing mode, but now I'm going to switch into my evolutionary biologist mode and think about what do we need to do to analyze this sort of sequence data. So the first thing is we have this sort of massive homology detection problem. So we have a whole bunch of these sequences. These are sequenced from lots and lots of these different trees, so we don't know which of the sequences fit in which trees. So let me just remind you that each one of these ancestors was created by one of these rearrangement events. And then each one of them that simulated some process of affinity maturation that generated a tree. But when we sequence, we don't, of course, know which sequences came from which trees. They just all got jumbled up in the sequencing. So this is the first problem, is we need to decide from this big pile of sequences, and when I say big pile, I mean like in some of our experiments we have 5 million unique sequences. We need to be able to divide them up into trees so that we can do phylogenetics and learn about the evolutionary process. So the way that we are approaching this is we build an HMM that basically is a likelihood model of how this BDJ process works, and we use it to decide whether pairs of sequences are homologous or not. So in this is a hidden Markov model. So we have states, categories of states, some for the B, some for the D, some for the J. And when we have a single sequence, we make a path sort of matching a given B gene, and then we go into a non-templated insertion state for a while, and we jump over to another gene and so on. So we can calculate the probability of observing a given sequence for a given gene. So now when we have two sequences, what we do is we form a likelihood ratio, which is what is the relative likelihood of taking two sequences and having them march along in tandem through a given path versus two independent runs through. So we take one sequence and then we allow it to go in another direction for the other sequence. So using this, I'm not explaining this in very much detail, but that's the essence of the idea, is comparing the likelihood of this joint emission versus the independent emission. So we can use this to build basically a clustering algorithm to solve this homology detection problem. And if you're interested in details about this, this is done with my postdoc, Duncan Ralph, who's actually the brother of Peter Ralph, so hopefully some of you know Peter. He's a lovely individual who's here at this conference. So that's the HMM detection problem. Here's another thing that's interesting about this setting is, again, we have this enzyme that's purpose-built to mutate our genome. And it has specific properties. So typically when we think about mutation, it's not totally crazy to think that mutation happens somewhat uniformly across the gene. But here it's definitely not the case that mutation happens uniformly across the gene, because this enzyme, this AID enzyme, has specific preferences about where it likes to mutate. This is some nice structural work that was done just quite recently. So we're going to model this using per site F81 models. So every site gets a rate, and every site gets its own substitution matrix. And so this is, we have lots of genes with lots of sites, and so we get to a massive number of parameters. It's sort of absurd to try to fit this number of parameters without some sort of regularization. So what we've worked on so far, and this might not be the final way in which we do this, but the idea is to take a multiple sequence alignment of the germline genes. So therefore they come in columns. Each column has some prior parameters for a gamma distribution, and we assume that each mutation rate for each site is a drop from that gamma distribution. And this allows us to pull together the parameters for homologous sites, and we get much more effective parameter fitting for this situation. So I'm going to show you a couple plots. What I'm going to show you is, for these various gene families, I'm going to look at what the mean of this gamma distribution is, which is sort of some aggregate measure of what is the substitution rate at the various sites for the different gene families. So here's one such plot. This is for a family called V1. And there's not a whole lot to take away from this so far, except to see that this is a log scale, and so we're seeing order of magnitude variation in the substitution rate at neighboring sites. So now here are two other commonly used gene families. These are V gene families, and we can see they bounce around, but I haven't said anything so far that convinces you this isn't just big noise. So I hope to do that next by taking an individual, so a totally separate, unrelated individual, a different blood draw, and then superimposing that same plot. So now we're going to have two purple lines and so on, and then I'm going to fill in a region between those two plots. So that comes out looking like this. So the thickness here represents the difference between the two individuals, and we can see that even though there's a lot of difference between the gene families, sometimes, say here, the difference between the two individuals is rather small. So I interpret this as saying that there's a whole lot of consistency in this mutation process. Mutation and selection process in this case. Okay, so that's just looking at substitution process. This is work that I'm writing up with Vladimir Menin. So now let's do a little bit of natural selection. So I'm sure many of you are familiar with the fact that typically for these sorts of studies, we like to compare the number of synonymous versus the number of non-synonymous mutations, with, for example, we see an excess of synonymous mutations, we interpret that as being purifying selection. So this is made a little bit trickier by the fact that in antibodies, because of this mutating enzyme, there are certain types of mutations that are more likely than others. So, for instance, there's these certain biases in terms of the context. So this interacts in a complicated way with this process, right? I mean, there are these two different things. So it turns out that there's basically an inline control, and I'm not going to have to go into this, but there's an inline control with this process where we can see this sort of mutation without the effect of selection. And we can use that to infer a per site natural selection estimate. So there's part of the V gene that forms basically the stalk of the antibody, and that's called the framework region. And with immunologists, they typically think of this as being somewhat fixed. But here, if we're looking at the level of purifying selection, we can see that it varies quite a lot in this so-called framework region. And then the neutral, we can also detect neutral evolution and also diversifying selection. And so there's cells that are undergoing diversifying selection in this framework region. And again, it's nice to compare between individuals and see that we see a remarkable level of consistency between individuals for these natural selection estimates. So this is work that's already published, and that was joint with McCoy, Trevor Bedford, and Blatterman. And I would just, as a final note, I would remind you that all of this arose via evolution. So that's just sort of astounding to me, and something that I hope to think about more in the future. So I'll stop there and take any questions. Thank you.